Okay, so this is part two of the Unit 2 Lecture 1. So the first video for this lecture covered basically some information about cancer through mitosis and ended off talking again about cancer. And now we're going to pick up right here in your lecture outline talking about meiosis. And so that's where I'll bring up our PowerPoint presentation here. And so meiosis is really the other type of cell division we're going to deal with, and it deals with making cells called gametes or sex cells. So this pretty cool image here shows you this really large, it looks like it's orange in color, cell. So that would be an egg. That's one type of sex cell that comes from the female, and then the male makes a sex cell called the gamete, and they're colored kind of yellowish here. There's quite a few of them. They're quite a bit smaller than the female gamete, the egg. And so this process called meiosis is all about taking a cell and dividing it and making cells called gametes or sex cells. And so basically both males and females do this process. In the male, this process happens in the testes, and in the female it happens in the ovaries. So there are cells that go through this division process and basically make these gametes or sex cells, and that process of doing that is called meiosis. So if you remember from previous lectures, we mentioned how many chromosomes are in one of our body cells in a human, and the answer to that was 46 or 23 pairs, so 46 altogether. So if an individual has 23 pairs of chromosomes in all of their body cells except their gametes, then when the gametes fuse, so when fertilization happens, in other words, when the sperm fertilizes the egg, then that results in one cell called the zygote having 23 pairs or 46. So actually each one of these gametes is going to carry 23 chromosomes, and when they fuse, these gametes fuse, that cell is going to have 23 pairs. Each one carries 23, and then the 23 from each pair up, and now we have 23 pairs or 46. So basically this process called meiosis is all about taking a cell that has pairs of chromosomes and dividing it, and dividing the nucleus in that cell, and making cells called gametes that don't have pairs anymore. They just have 23 chromosomes. So you can see in the image here it says unpaired, and both a sperm and an egg. So that's what we're going to get into and talk a little bit more in detail about that. So this is showing you a couple images. On the left we have what's called a karyotype, and a karyotype is basically what they do is they take all of the chromosomes out of one body cell, and they put them under a microscope after they pair them up, and that's what we see. So here we can see there's 23 pairs, 1 through 22, and then the 23rd here is showing you the sex chromosomes, and we'll talk more about those later on. But anyway, take the 22 here, and then like this pair here, and then we have 23 altogether. If it's a female, then there's going to be what's called 2X sex chromosomes, and if it's a male, then there'll be 1X and a Y, much shorter Y chromosome for a male. So that's kind of what this image is showing you. For a female, you'd have 2X's versus a male having 1X and 1Y. But really what I want to show by looking at this slide is what's called homologous chromosomes. So remember, your body cells, every cell in your body, except your gametes, if it has chromosomes, those cells will have pairs of chromosomes. And so why are they pairs? Well, remember, you get 23 chromosomes from the sperm, from dad, and 23 chromosomes from the egg or mom, and then they pair up, and now we have homologous chromosomes. So we can see there's the first pair is pretty long, and then as we go through, they get shorter and shorter, and we get the shortest down at the end here. Over on the right, we have a red chromosome and one that's colored blue. So these are homologous pairs, and so we could say, for example, that this pair is pair number 1. And we're just kind of drawing an animation of it here, an image of it. And so the red one, let's say, came from the egg, and the blue one came from the sperm. And when fertilization happened, now that cell that may turn into a baby has a pair of these chromosomes now. So homologous chromosomes are not identical. They don't have the same exact DNA, which makes sense because your parents are not identical genetically, right? So you get one chromosome here, for example, from mom and one from dad. They're not exactly the same chromosomes, but when they pair up like we're seeing here, pairs of homologous chromosomes have genes that code for similar characteristics. So, for example, this red one may have a gene for, let's say, eye color, and then this blue one could have a gene for eye color as well. And so they're not identical, but they're very similar in that respect. So let's say this eye color gene on the red one, it has a blue eye color gene, and let's say this eye color gene on the blue one has a brown eye color gene, right? So they're not identical genes, not identical pieces of DNA, but they're similar in that these chromosomes have genes that code for similar characteristics or similar traits, right? So not identical, these homologous pairs of chromosomes, but again they're similar and having similar genes, right? So how many pairs? Well, remember there's 23 pairs in all of your somatic or body cells, and so that's how many we have as humans. Remember, different organisms could have different numbers. So when we talk about pairs, we can use what we call the term diploid. So di, means 2. So when you have a cell that has pairs of chromosomes, then it would be considered a diploid cell. If you have a cell that has one member of each pair of chromosomes, that would be called a haploid cell, or a cell that has half of the DNA. So if you had a diploid cell, all these chromosomes would come out of a diploid cell because there's pairs. You have a pair here, number 1, a pair here, number 2, number 3, right? But if you had a cell that just had one member of each pair, so let me grab my laser pointer here. Let's say it just had this right chromosome of the first pair, and this left chromosome of the second pair, and this left one of the third pair, so only one of each pair, then that cell would be called haploid. It would have half of the chromosomes, in other words. So when you do this process called meiosis, and you make gametes, you're going to end up with these cells, these gametes, sperm and eggs, that have half of the chromosomes. They're going to be haploid, in other words. They're not going to have 23 pairs or 46. They'll have just 23. And then when fertilization happens, the resulting zygote that forms will have 23 pairs and will develop into a baby. So this is showing you another karyotype. This time this comes from, it says up here, a female. And you can see that the very last pair, whoops, let me grab that. The 23rd pair here, there's 2 X's, so that would make it a female as a result. So if there was a Y, 1 X and 1 Y, that would be a male, right? And actually that's what this karyotype is showing you. So these are all the chromosomes out of one somatic cell out of a male. And we know it's a male because there's an X and a Y chromosome here for the 23rd pair. Um, so again, to do meiosis, when you do meiosis and you make gametes, you're going to end up having cells that have one member of each of these 23 pairs. So it'll have half the chromosomes, and it will be considered haploid as a result. So we're going to draw it. Before we draw meiosis like we drew mitosis, a couple things that to consider. You have most of this in your lecture outline, but there's actually, it's pretty different from mitosis, but it's also very similar too, we're going to see. Unlike mitosis, meiosis has two cell divisions. There's what's called meiosis 1, which is the first division obviously, and then meiosis 2 is the second division. And so remember, mitosis just had one cellular division. You copied all the DNA during interphase. You made sister chromatids, in other words, right? And then you separated those sister chromatids, those identical chromosomes, and you ended up making two cells that were genetically identical to one another. Well, you actually do that here in meiosis during meiosis 2. You separate the sister chromatids just like you did during mitosis. But meiosis 1, that comes before meiosis 2, is a lot different. And we'll talk more about it. But basically, it starts out the same as mitosis. So you're going to have basically double the DNA, because interphase occurs before it happens. So you replicate every chromosome, and you have sister chromatids to begin with. And then something called crossing over happens, we're going to find out, during meiosis 1, and that's where chromosomes basically exchange genes with one another, pieces of DNA, and you get some genetic variation that results. And then you separate homologous chromosomes. So that did not happen during mitosis, but we will see that happens in meiosis 1. And at the end, you end up with two cells that have half of the DNA. In other words, you no longer have pairs, homologous pairs in each cell. You now have one member of each pair. Now you have haploid cells. But you still have sister chromatids, we're going to see. So let's draw it, and this will hopefully make a little more sense, and we'll watch a little video too. So we're going to start here with, let me grab my pen, come on, there we go. And I'm gonna, so we're gonna start here in interphase 1. So basically interphase, right, which happened before mitosis. Now obviously we're dealing with meiosis. So in interphase, remember, all the DNA, all the chromosomes, basically, is copied. So all the DNA is copied, and so we're gonna make sister chromatids. Right? We're going to make identical chromosomes in interphase. So I'll just write that like we did in the mitosis drawing. So now we have double the DNA, right? So we're going to begin just like we did with mitosis, with double the DNA. So so basically, remember, we have meiosis 1 that occurs, and then it's followed by meiosis 2. So this drawing is showing you so we have a cell here that represents prophase 1, and then another one that represents metaphase 1, anaphase 1 here, and then we end up at the end of telophase 1 with two cells that are haploid, that have half the DNA. And then meiosis 2, which consists of prophase 2, metaphase 2, anaphase 2, telophase 2, is going to take these two cells that we produced in meiosis 1, and each one is going to divide into two others. And so this one will divide into these two, this one will divide into these two, and we will end up with 4 haploid cells at the very end. And so meiosis 1 ends up with these two, meiosis 2, we end up with 4 at the very end. So we start with 1 cell that's diploid, that has pairs of chromosomes, and we end up at the very end making 4 genetically unique, not clones, haploid cells. We'll find out. So in prophase 1, what happens is, just like in prophase of mitosis, the chromatin, remember the uncondensed DNA, chromatin starts to condense into visible chromosomes. And remember, we have double the DNA, so we have sister chromatids that happen. So I'm going to, again, just simplify it a whole lot and pretend that we're just dealing with one pair of chromosomes. But remember, in us, we would have 23 pairs, all doing what I'm going to show you here. So I'm going to draw one chromosome right here. But remember, there are sister chromatids, so there's going to be an identical copy, and they're connected by that protein structure called a centromere. And we're not just going to have that one, but we have pairs, remember? So I'm going to draw blue to, represent one that came from the sperm. Let's say the red came from the egg. And so here we have the one that came from the sperm. But remember, there's identical copy, a sister chromatid connected by a centromere. Okay, so we start to see those in prophase 1. And what else happens in prophase 1 is that these homologous pairs end up coming into contact with one another. That did not happen in mitosis. So I'm going to actually erase one of these here. Let's see. Let's do this. I'm going to erase the blue one, and I'm going to draw it closer to the red one, because that's one key characteristic of meiosis 1, specifically prophase 1, is these homologous pairs basically come together and kind of touch each other at certain areas. And the areas that they touch, they will basically exchange DNA with one another. So this red one here, now let's say, has a little blue on it from the blue one, and the blue one now has a little red on it. And so now we have these recombinant chromosomes, in other words, or chromosomes with DNA from both mother and father on it. This process that we just talked about and I just drew is called crossing over. And so crossing over is where these homologous pairs of chromosomes cross over with one another and exchange DNA with one another. And so that gives us some genetic variation. Now these chromosomes are different genetically because of that. And so that adds to the genetic variation that's produced in our gametes, in our sperm and our eggs. Okay, so that is basically prophase 1. That's the major idea of what happens there. So then in metaphase 1, what happens, just remember in metaphase of mitosis, very simple, right? Remember the sister chromatids went in the middle of the cell, or the metaphase plate we called it. Another term was the equatorial plate of the cell went into the middle. So that's basically what happens here. These homologous pairs, remember, that are basically together, will come and get right in the middle. I already kind of drew them in the middle above, but I'm going to draw them again here and put them directly in the middle. And remember, some of the red now has some blue, like we drew up above. And some of the blue has some red on it due to crossing over that occurred. So that's metaphase 1. They line up in the middle. Anaphase 1, remember in anaphase of mitosis, what happened? Well, if you remember, what happened is the sister chromatids separated from one another. But that does not happen in anaphase 1 of meiosis. That happens later, we'll see. But what happens in anaphase 1 of meiosis is these pairs separate. So the red, remember that has a little blue on it now, separates from the blue. and we separate these homologous pairs from one another. And so let me draw. Well, I'll just draw it down here. So we have this going on, right? This one will be going that way, and the blue will be going this way. Not the greatest drawings, but you get the idea. And then remember, this has some blue on it here, and this has some red on it. Voila, there we go. And I'm going to draw an arrow representing the movement. So this one was moving to the left. Terrible arrow, but it's moving that way. That's moving that way. There we go. So that's anaphase, right? Basically the separation of them. And so now in telophase, just like in telophase 1, excuse me, just like in telophase of mitosis, we start to unwind these chromosomes back into chromatin, and we start to get basically cells that look like it has they have nuclei in them, right? And so let's see, I'm just going to draw this here. So again, we have a nucleus here with a nuclear membrane around it, and we have this red here that has a little blue on it, and I'll draw that in a second. And that blue would be in this cell's nucleus here. So there's a little blue on that, like we had over here and over here and over here, right? And then this blue again has a little red on it. There we go. Okay, so now we have at the end of meiosis 1, after cytokinesis happens, also dividing the cytoplasm, we have two cells that are now haploid. So remember, the cell we started with was diploid. It had pairs, homologous pairs, red and blue. Well, this cell here just has red, but remember, a little blue on it now due to crossing over. And this cell here just has the blue on it. And remember, it has a little red due to crossing over. But the basic idea is we do not have pairs anymore. Those pairs have been split, and we have cells now that have half of the DNA as the cell we began with. So we went from a diploid cell with pairs of chromosomes to haploid cells with one member of each pair of chromosomes. But what do we have in each of these cells still? This cell still has sister chromatids. You have basically one red and another red, although they're not really literally identical anymore due to crossing over, but we still call them sister chromatids, and we need to separate those in order to get genetically compatible gametes. So we need to separate the reds and the blues and make cells that have only one of those chromosomes. You don't want double the DNA in a gamete, right? So just like in mitosis, we separate sister chromatids. So it's basically the same exact thing that happened in mitosis, except now obviously we have cells that are haploid. So at the very end of meiosis 2, after we go through prophase 2, where the sister chromatids start to condense, metaphase 2, they line up in the middle. Anaphase 2, we separate those sister chromatids. In telophase 2, again, we start to see a nucleus in the cell, a nuclear envelope or a membrane with the chromatin inside of it. And so just to kind of draw that here, I'm going to draw a circle to represent a nucleus in each of these 4 cells. So there's a nuclear envelope, and grab my red. So this nucleus will separate, and one of these will go into this cell, and the other one will go into the other one. But remember, one of them had some blue on it due to crossing over. So let's say this one up here had the blue on it. That's not great, but anyway, there's blue on this, right? So one of these has some DNA from the blue chromosome. And then same thing with this one. So this pair will separate. You'll get one there, one here, and those sister chromatids separated. But remember, one of the blues we drew some red on, so I'll draw some red on this one here, and there we go. So again, we had sister chromatids here, but this cell was separated, and now we have one of the sister chromatids in this cell and the other one in this cell. And so at the very end, we end up with 4 genetically unique cells. If you look at them, none of the chromosomes are identical. So this has all red in it, you can see. This one, it has red with some blue in it. This one's all blue. This one, it has mainly blue with some red DNA in it from the maternal chromosome. So that is one key characteristic of meiosis, is making these genetically unique gametes, and we end up with 4 haploid sex cells, either sperm if we're talking about a male, or eggs if we're talking about a female. Okay, so we are going to watch an animation, and then we will talk more about this. Let me grab, let's see, let me grab this one here. There we go. So let's watch this animation. I'm going to...
The cell prepares for meiosis.
I'm going to back it up. I'm going to stop it a couple times as we watch it, but let's watch the beginning of it.
The cell prepares for meiosis just as it does for mitosis.
And that basically means that the cell duplicates its DNA. So we have sister chromatids before mitosis occurred. We have sister chromatids before meiosis 1 happens, right? And so we have double the DNA. Each chromosome was copied.
But as the DNA condenses, each chromosome finds its homologous counterpart.
And that did not happen during mitosis. So in meiosis, remember prophase 1 specifically, of meiosis 1, the homologous pairs pair up. They kind of come into contact with one another. So you can see here's a blue. This would be one of the sister chromatids. Here's the copy, another blue one. And then you can see the red. So these that red and blue pair are coming together. Here's another pair coming together, and up above there's another pair. So those pairs pair up, and that's unique to meiosis 1.
These matching pairs of chromosomes hold equivalent genetic information,
but not the same, remember? And so they're similar, the homologous pairs, in having genes that code for similar characteristics, but they are not genetically identical, the homologous pairs of chromosomes.
One set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The 2 homologs trade genes in a process called crossing over. This forms new hybrid chromosomes.
Okay, I'm going to stop it there, and you can see now this blue has some red on it from the maternal chromosome, and this red has some blue on it from the paternal chromosome. And so we crossed over, and now we have basically what we call recombinant chromosomes, or chromosomes that are genetically different now because of that.
The spindles then place maternal and paternal chromosomes randomly on each side of midline.
So the spindles are these white lines they're drawing here. Basically, spindle fibers, we've called them before, they push and pull the centromeres of those homologous pairs of the sister chromatids really into the middle, and as they do that, they line up randomly. We'll talk about random alignment in a second, otherwise known as independent assortment. So that's what the video is referring to, but don't worry about that right now.
It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart.
So that you just saw was anaphase 1. It separated the homologous chromosomes, or in the video they're calling them homologs. And so now we're seeing a cleavage furrow form on both ends. Cytokinesis is occurring, and you'll see telophase 1 happening, and so we're going to end up with two cells that are now haploid. We no longer have cells that have pairs of chromosomes, although that each cell will still have sister chromatids that will need to be separated during meiosis 2. But we'll finish meiosis 1 and you'll see that.
This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two.
So we need a second meiosis, basically, with meiosis 2, that separates sister chromatids, just like what happened during mitosis.
The second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Okay, so we end up with 4 genetically unique haploid cells at the end. Either sperm, they will be, or eggs. So that basically summarizes that process, and I will try to find another video too to upload to our this week's folder, so you can view a different video of meiosis as well. Okay, so just to summarize again, this is showing you meiosis. We start out with a cell that went through interphase, and now we have double the DNA. We have sister chromatids, and in meiosis 1, we separate these homologous pairs, right? So prophase 1 happens first, crossing over occurs. We get some blue on the red, some red on the blues here, and in meiosis 1, specifically anaphase 1, we separate those pairs, the blue from the red, and we end up with 2 cells at the end of meiosis 1 that still have sister chromatids, and meiosis 2 is all about separating those sister chromatids and ending up at the end with 4 haploid genetically unique cells. So that basically summarizes meiosis. So how do we get genetic variation in the gametes? Well, we talked about one way already, and we mentioned another one. So we talked about crossing over, right? And we'll see that is going to be 1 out of the 4. But another one is just random genetic changes that can occur. So those are called mutations. If you have your DNA that mutates or changes, that can happen, and that could lead to a sperm or an egg, or both, having slightly different genetic material in them, and so a mutation can occur. Another way that genetic variation arises through a process called random alignment. So focus on this top upper left cell first. So you can see that they're simplifying it here because they only have 2 pairs in our drawing, which is your 1 pair, right? Blue and a red. But now they have 2 pairs that they're talking about here and that they drew. And so if the chromosomes, homologous pairs, when they line up in the middle during metaphase 1, when they're pushed and pulled in the middle, you could have them align like this in the middle. You could have this pair line up like that with the red on top and the blue down below. There's another pair over here where it's the same in that you have the maternal or red pair above and the blue below. And if they lined up like that, then the blue would be separated and you'd get end up with all blue in the sperm or the eggs, in 2 of them. And then if the red were above all of them, then you would end up with sperm or eggs that had all red in them, right? So that's one way that the homologous pairs could align. But they could also align just as equally like this. So remember, in the scenario above, you had the reds all above. Well, here you have one of the red above and then the blues below. But over here, one of the red is below and the blues above. And as a result, when anaphase separates those homologous pairs, you're not going to have all blue and all red. Now you'll have basically some blue and some red in each resulting cell. So that's called random alignment, as to where when they align in the middle during metaphase, it's just random to how they align. And that can lead to genetic variation as well. So that's another way. Another name for that you might see in your textbook is independent assortment. But I think most of the time you'll see the term random alignment. So that's another way. Another way is through what we call random fertilization. And I am gonna... Okay, so I had to just pause that real quick. Let's get back here. So mutation, genetic mutation, random alignment, or independent assortment. Random fertilization basically means any one sperm fertilizing any one egg. So in humans, each male and female gamete represents one of about 8.4 million possible chromosome combinations due to random alignment. And the fusion of any one male gamete and any one female gamete during fertilization will produce a baby with any of about 70 trillion diploid combinations. So, saying that we are all unique is not an overstatement, because we are very genetically unique because of that. So, but another to add into the possible combinations, which makes it a mind-boggling statistic to think about, is crossing over. So remember when we had some of the paternal or blue, we're coloring it here, exchange with some maternal red, that's crossing over, and you get recombinant chromosomes. You can see that term down at the bottom of the figure, that result. And so that just adds into the number of different possible zygotes that can occur when any one sperm fertilizes any one egg. So all four of these, mutation, random alignment, aka independent assortment, random fertilization, and crossing over, all add to the genetic variation that is produced in us. And so it's a pretty mind-boggling scenario. So I like to, when I talk about this in face-to-face courses, I like to give this scenario. So if you took the same parents that produced you, so your mother and father, and they make another baby, a sibling, in other words, there is a very, very slim, if no likelihood at all almost, that your sibling would be genetically identical to you. I like to say at least a 70 trillion chance that your sibling would have the same chromosomes as you do. So all these are involved in making us all genetically unique, basically. So there could be a problem that happens in meiosis, and it's called non-disjunction. Non-disjunction is basically a process that leads to cells having abnormal chromosome numbers. And you can see it happening here in meiosis 1 in this figure. So here's a cell, and we're supposed to separate the red from the blue, right? The homologous pairs during meiosis 1. Well, you can see what's happening here is the pair on the right is separating like it should, but the pair on the left isn't, and actually both members of that pair are going to the same side of the cell, and as a result we end up with cells having too many chromosomes and too few chromosomes, and that can lead to a gamete having too many or too few chromosomes. And so there's a lot of different conditions out there that result due to non-disjunction we'll talk about. Here's a video. I'm going to show you just I'm going to shut off the volume here and increase it. And this video is going to show you non-disjunction happening. So let's watch the first couple seconds of this video.
This newt cell turned out to be especially interesting. One of the two poles is shown by the circle, and some fibres attaching chromosomes to this pole are just visible at the arrow. During metaphase, two chromosomes get caught up on the wrong side of the pole. One manages to get to the middle of the spindle just in time for anaphase, but the second doesn't make it. Manages to get to the middle.
Wind it. So they're showing you all these chromosomes are basically lining up in the metaphase plate, the equatorial plane, and this chromosome, the sister chromatid, doesn't get over here in time. And so what happens is it should end up being split, but it doesn't get over here, and so the cell that will result with all these chromosomes will have too many, and the cell on the right-hand side would have
Middle of the spindle just in time for anaphase, but the second doesn't make it. As a result, the two new cells will die, because one cell has a double copy of this chromosome, while the other cell misses out.
But sometimes they don't die. In other words, you could have a gamete that is just fine, but it just has too many or too few chromosomes, and that could result in the baby having too many or too few chromosomes. And so we're going to talk about a couple conditions here that can result due to what's called non-disjunction. So Down syndrome, also known as trisomy 21, due to having one extra number, the 21st pair of chromosomes, and that results in a condition called Down syndrome. And there are, certain characteristics that individuals that have trisomy 21 have. So they have a characteristic facial appearance, some form of mental impairment. Sometimes their individuals are sterile and not capable of producing children themselves. The incidence of this increases with the mother's age, they have found, and so once the female gets to about 40 years of age, the incidence of having this non-disjunction in an egg tends to increase for some reason. So that's what they have found in the recent past. So there's other non-disjunctions as well. Trisomy 21 is, very well known and pretty common one. Turner syndrome is where there's just one X. So if it'd be a female with not two X's, just one. Something called trisomy X, where you have three X's instead of just two. And Klinefelter's is where you have it's a male that has two X's and one Y. So too many chromosomes there. Trisomy 21 we talked about. Down syndrome. Patau is where you have one extra number, 13, the 13th pair, you have one extra. So there's quite a few other conditions that result due to non-disjunction. Okay, and that the very last thing I'd like you to do is compare and contrast these two processes, mitosis and meiosis. So we talked about mitosis in part 1, and we ended off talking about meiosis in part 2. The green is showing you mitosis. The kind of purplish color on the right side of this image is showing you meiosis, and kind of visualizing that. So I would like you to compare and contrast these two, and you might see this on your exam as well. Hint, hint, right? So I'm going to do this. I'm going to draw two circles. You might have heard of something called a Venn diagram before, and Venn diagrams are useful in compare-contrast scenarios. So two circles, and both circles are going to kind of overlap one another. And so actually, you know what? Let me do this. I'm going to draw one circle black here, just to differentiate them, and then the other circle I'll draw in red. So there are two circles overlapping. This red one, let's say this is the meiosis circle. And the black one, we'll say, is the mitosis circle. So anything that is unique to mitosis, you would write just in the mitosis area. Anything unique to meiosis, you would write in just the meiosis area. And then anything similar between the two, you would write in this area where they both overlap one another. So I'll give you a couple examples. So one, let's use just blue here for general. So for mitosis, basically you result in how many cells. Do you remember how many cells are produced at the end of mitosis versus meiosis? Mitosis, you end up with basically genetically similar clones. So two cells that are genetically identical. And I'll just put in parentheses clones. So they're cells that are clones of one another, genetically identical cells. In meiosis, you ended up with 4, remember, at the very end, 4 genetically unique, I'll just put unique, so not clones. It's hard to write with this mouse here. 4 unique gametes, right? So you make sex cells, and there's 4 genetically unique sex cells or gametes at the end. So meiosis 4, mitosis 2, genetically unique in meiosis, similar clones in mitosis. What are some similarities between the two? There's quite a few of them. Both of them separate sister chromatids. So you separate sister chromatids in mitosis, and then in meiosis 2 you separate sister chromatids as well. Sister chromatids. Write it here. Terrible writing, but I think you get the idea, right? Chromatids, the identical chromosomes, remember, sister chromatids. And in meiosis, there was one other division, right, that mitosis didn't have. So what did we separate in meiosis 1 that we did not separate in mitosis? It was homologous chromosomes. So we separate homologous chromosomes in meiosis 1, but we don't separate them in mitosis. So you could write separates, and I'm not going to write it here, but I would definitely write it down so that you can use it to study for your exam. Separates homologous chromosomes in meiosis. Another difference could be crossing over. Did crossing over happen in mitosis and meiosis, or just one? Just happened in meiosis, specifically meiosis 1, and where we basically exchange DNA with the maternal and paternal chromosomes. And so crossing over happened in meiosis, but not mitosis. Both of them, you could write before both, or even just in the middle, so something that they both share. Interphase copies the DNA before. So we have sister chromatids before mitosis, and we have sister chromatids before meiosis. So that's something they both share too. So think of some more things, and you may see something like this again on your exam, where you'll have to come up with some unique features of both of them, and some features that they have in common. Alright, so with that, we are done with part 2, and I will see you when we get to the next lecture.